So welcome everybody. Today's topic is computational approaches in clinical epigenomics, and it is being presented by Dr. Fei-Man. She is a postdoctoral fellow and IDRE Fellow at Pellegrini Lab in the Department of Molecular Cell and Development Biology at UCLA. With a PhD from University of Tokyo, Dr. Shu specializes in bioinformatics. She holds a MS degree in molecular and cellular biology and BS in life sciences from National Taiwan University. So without much ado, let's welcome Dr. Shu and Dr. Shu.
Thank you, TV, for the introduction. So can you hear me well?
Yes.
Okay, thank you. So hello, everyone. Thank you for joining my presentation today. So today I'm going to describe some computational approaches in clinical epigenomics. So I'll go through every method with an example in our recent studies to study the virus host interactions with DNA methylations. So yes. So first, so that's I'll briefly introduce the main role that we are studying today first. We are studying a virus called cytomegalovirus. In short, for CMV, most of the reports or website you will see. So from the name, you will know the features for this virus is it has a large, more than it's a size bigger than the average of the virus we see. So in this graphic, you will see like the cytomegalovirus, they are enclosed in an envelope, and the diameters is around 220 nanometers, which will be like 4 to 10 times greater than the average of the regular virus we see. So cytomegalovirus, that belongs to the herpes viridis, which is a subfamily like called beta herpes viridis. So cytomegalovirus, they will also infect many of the human cell types, including fibroblast, blood cells, and even hematopoietic cells, epidermis, etc. So it contains a double-strand DNA in its genomes inside this capsid. So one of the features that when we can test if this person is infected by cytomegalovirus before is from the histologies, you will see this specific features they called inclusion bodies. Usually they show up in pairs. It shows like features like an owl eyes. So these inclusion bodies, they will contain high concentrations of the virus particles, either in the nucleus or in the cytoplasm. So cytomegalovirus came into our studies because in fact, more than 60% and even in some areas to even to 100% across the world, I mean, among those adults. So the transmission is basically through body fluid, like blood transfusions, etc. So the infections rate also depends on the hygiene conditions, how development for that specific area is. And one type of infections is called congenital infections, which means the baby or the fetus, they will be exposed to the virus when they were in the mom's uterus or during the phase during deliveries. So past literatures found like cytomegalovirus congenital infections cases, these babies during the growing up, they might develop into some problems, for example, seizure or developmental delays. They might have a bit vision problems. And the most phenomenal one would be like they might lose their hearing abilities. So you can see they damage their neural systems. So one of the reasons like those babies, they become with defect is because they exposed this to the virus really early, and their immune systems may not be established well during that times. So they will have this obvious defect showing up later in their life. But most of time, people, when they were adult, their immune systems are built. When they encounter the cytomegalovirus, the virus usually just stay dormant inside the bodies. They call latent infections, which means the virus just sit there, like showing up here, and without causing much problems to your body. It will become active or revive when this person, they got some problems like immunocompromised. The most significant case would be like people getting treatment for chemos, or when these persons, they have to do surgeries, for example. The most phenomenal one would be organ transplant. So when they have to take another organ from other individuals, after the transplant, they might go through immunosuppressions to avoid the rejections for the organs from the other person. So this is the time like CMV came to the games. So they will take the opportunities and attack the host immune systems. This time, the latent virus, they will back into probably lytic phase, which means the CMV reactivated and starting to replicate their genomes and producing the virus particles and take every usage of the host resources to propagate their own genomes. So this is the target we're going to study. We're trying to study the epigenetic mechanisms between the CMV pathogenesis in the host epigenome. Okay. So the epigenetics is a mechanisms like they will modify many of the biological processes without changing the DNA sequence. So DNA basically composed by the 4 bases, A, C, G, and T. You can see this infograph is like epigenetics. They have different kinds of. They have histone modifications. They have chromatin accessibilities, like nucleosome positioning, etc. But our main focus today would be DNA methylations, showing up here. So DNA methylations means adding one of the methyl group to the 5th carbon of the cytosines, which is one of the DNA nucleobases. So this adding methyl group that will be done by a group of proteins called DNA methyltransferases, in short for DNMT. And these DNA methylation process could also be reversed or oxygenized by another group of proteins called 1011 translocations, or TET. This would be an examples in human and in mouse, like mammalian cells. In other organisms, they might have different group of proteins to do this process. And DNA methylations is heritable, and they will be involved in several biological processes because they might modulate gene expressions. You might heard of imprinting. It's like when individuals in their body, whether this gene is regulated, is coming from imprinting signatures from their parental side or maternal side. And DNA methylation in the past also been recognized to be able to silence some virus replications and also like transposable elements, like when retrovirus integrate into like human genomes that become inactive because they be highly methylated to let these transposable elements to be active. But given though there are several studies about the DNA methylation and virus silence, that we have very limited knowledges for the role of DNA methylations in the CMV pathogenesis we just talked about before. So that's why we have these studies. So this is so far what we know about some epigenetic mechanisms in CMV reactivation. is mainly focused on histone modifications, but not DNA methylations. For here, you can see in this graph, there are several circles in red or green shown here. So these are histones. So you see there's a group of promoters, that in short for NIUP, which is a major immediate early promoters. So they will show different features when CNV virus is inactive, which means latency, or active lytic phase. So these histones, they become more condensed to each others. So they might be, because the chromatin will be less accessible and become inactive and more enriched with some chromatin repressor complex. So in this stage, the immediate early group of genes, the lytic phase genes, will be silenced, which means the downstream genes will not be active. But once the virus become reactivated, these group of promoters, you can see their histones become more loose. So these give other proteins, transcription factors, the chance to binding onto these regions and then trigger the downstream gene expressions with RNA polymerase II. So you can see this type of proteins would include NF-kappa B, AP1, CREB, this kind of proteins we have been known for long, involved in inflammation, DNA damage, leukemia, reperfusion injuries, etc., which means when the host under stress, the CNV will become reactivated. So this is the mechanisms we know in CNV reactivation with some histone modifications studies like K9 trimethylation, K27 trimethylations. But we want to know about DNA methylation's roles inside this process. So the first questions we want to ask is there any association between DNA methylations and CNV serostatus, or in other words, the CNV infections will cause any changes in DNA methylations. Okay. So we have the study designs showing up in this graph. This one would be a clinical studies that we recruit human subjects. So we have two cohorts. One is a group of people, they are about to get kidney transplant in UCLA, and another group will be, they are about to get lung transplant in UCSF, which give us a good opportunity to study the CNV and DNA methylation relationships, because we know for transplant patients, they will be about going through immunosuppressions after the organ transplantations. Okay, so we collect the blood from these patients at the day they are getting the organ transplant surgeries, and we collect the post-transplant samples after 3 months of the transplantation for the kidney cohort, and 1 year after those people getting from the lung transplant. From these blood samples, we isolate peripheral blood mononuclear cells, which will be a thin layer showing up here. So they will be enriched for those white blood cells, immune cell types. So for these biopsies, we put them into targeted bisulfite sequencing. We'll introduce this really quick in a few slides. So this is the way we profile their genome-wide DNA methylations. So finally, we'll get the sequencing data, which including methylation call, and also we can get some ancestry informations from the patients, and also we can profile their cell type compositions from the methylation data we got. And together with the information we gathered from the clinical side, we will get their demographic records, including wide range of age, different sex, and the most important part would be their CNV serostatus. For all the features, we're modeling these features with methylations into a model, so we can study in which specific loci like DNA methylation contribute to the CMV infection. Okay. So let's give a quick look at the study cohorts information. So we have in total 156 or so pre-transplant samples coming from kidney and the lung cohort. There will be variations in ages and sex, and we also recording their ethnicities. Most important part would be these patients, we recording their own CMV serostatus, which means whether they have been infected with cytomegalovirus, which would be the recipient. And they will be given an organ, which from the donor, they might have different serostatus coming. So we have these records for all of the patients. And some of them, after they got transplanted, we also sampled for their post-transplant blood samples to profile the DNA methylation. So once we got this and we collect their PBMCs and perform the targeted bisulfite sequencing on these hundreds of samples. From the names, you will know targeted means we are sequencing only those targeted regions. Just like the regular bisulfite sequencing libraries, we got the DNA extracted, sharing them, put on with the Illumina sequencing adapters, multiplex them together. But after this, we put one more step, which will be called hybridization captures. In this step, we put in probe, which will be the single-strand RNA labeled with biotin. So this probe will hybridize with the regions complementary to them. And once this hybridized, they can be going down through the bisulfite conversions. We'll talk about this later. So to measure whether the cytosine is methylated or not. Okay. So after this, we sequence them, get the data, and trying to analyze this. So you can see this step, the hybridization capture part will be the key for the TBS-seq. So we will design probe panels, which will be the region of interest across the human genomes that we are trying to look into. So this probe might be related to any health outcomes like diabetes. They might be related to some accumulative traits like whether these people have gone through smoking or so. And they will have higher intra or intertissue variances, and they might be correlated with the age changing. Okay, so with this method, we efficiently cutting down all the sequencing amount we need for one individuals. So remember, we got more than 100 clinical samples. We have to do the sequencing. If we wanted sequencing the whole genome for humans, per person, there will be 3 GB multiplied with the coverage. So it will be way too much. So we use targeted bisulfite sequencing with the panel, which is sequencing for 4 million bases per individuals. And we can give every individual enough coverage to analyze this size of methylation. Okay. So let's talk about bisulfite sequencing, which has been developed for decades for us to acquire cytosine methylation in single base resolution. You can see here, there's a DNA sequence shown up here with the cytosine to be methylated or unmethylated labels. For this sequence, we treated them with sodium bisulfite in high temperature and enough long time. This process will call bisulfite conversions. So the methylated cytosines after these bisulfite conversions, they remain to be seen in the readout. But for those unmethylated cytosines, after the bisulfite conversions, they will be converted into thymine, into T. So this is the readout we get by comparing to the reference human genomes, we would know specific locations, whether it is methylated or not. And we can do this by a package developed in our lab. It's called BS-boat. So they can do the alignment for those bisulfite reads to the human reference genomes. And after these comparisons for each loci, we'll accumulate enough read counts, and we can get like the percentage or the ratio for methylated C or unmethylated C at the specific positions. And we can do this with the methylation call also in the BS-boat package. So this is the way we measure for per base methylation levels. So that's so we've introduced the study concept behind and how the technical side, how we measure methylations. Now let's get into the data. Is how the first question we want to ask is there's any correlations between methylations and the CMV infections. So we are profiling the pre-transplant samples here because this would be a less biased populations without any treatment, any surgeries, and any immunosuppression prophylaxis involved. So for these targeted bisulfite sequencing samples, every of them will acquire more than 30K of CpG size to analyze. So from these metrics, we trying to see if there's any correlations with the traits we're interested in. So like expected, we see the methylation is highly correlated with the main traits will be the sex. So you can see the populations will be grouped very well with the sex. But among this female cohort, you can see there's a slightly correlations across the UMAP1 axis, which like CMV negative patients going to the right, but CMV positive patients is more clustered to the left across these axes. So this tells us there might be the minor or subtle correlations for methylations to the CMV serostatus. Might not be prominent like the other accumulated traits, but it is there. The next question we would like to tackle is, since this is a clinical studies, we have to take advantage of what types of biopsies we got. So from these studies, we, like I said, we get the peripheral blood mononuclear cells, which would be a group of white blood cells inside the whole blood collected. It should be the thin layers after centrifugations. So in other terms, is the methylation call we got would be a composite variables coming from all of the cell types. And we can actually deconvolute every cell types inside this liquid biopsies based on the methylation call. So we assume the PBMC samples from literatures mainly come from the lymphoid lineages in the hematopoiesis. So T lymphocyte, B lymphocyte will be the two main contributions to the final PBMCs. There will be a little bit nature killer cells and very low percentage of monocyte included as well. For other granulocyte, there should be depleted or in a very, very low concentrations inside these PBMC samples. So we're trying to deconvolute these cell types with a models. So imagine like PBMC methylome, the final matrix we got is composed by several group of cells. So which will be the Y response variables inside a non-negative list regression models. So this is the way we're trying to do deconvolution. So here we are trying to get the argument min from these list regression problems. So Y is our final PBMC methylomes. A would be a matrix, assuming it's coming from different cell types in row, different methylations in columns, which would be the A matrix. X will be the things we're trying to derive, which would be the coefficient vectors per cell types. Finally, we'll optimize this to get a argument minimum from these non-negative least regression models. So from here, we know that the first thing we need to do is we have to build a reference database to do cell type deconvolutions. And we going through literatures, we know there might be CD8 T cells, activated one, CD4 T cells, naive T cells, B cell, etc., and also NK and monocyte. There might be very few immature granulocyte. We're lucky to find a neutrophil band database from the blueprint database. So these data from these reference cell types, we do one versus all comparisons to isolate specific DNA methylation signatures, like shown up here. So you can see in x-axis, these are differentially methylated regions for each cell types. So with these signatures, we can put them into the model and estimate the coefficient for our final PBMC methylomes. After we build model, we even do synthetic mixtures to verify the max absolute error to see if this model is actually giving us very low error rate. And the answer is yes, we can get an error lower than 5% for some, even 2% for some cell types. So we run through every samples to estimate the cell type composition and find out there's one cell types which would be the activated CD8 T cells to show statistical differences between the CMV negative and CMV positive group of patients. For the other cell types, they remain similar without changing. Okay. So brief summary for now. We do general profiles to do dimensional reproduction with UMAP, and we see some minor association for CMV seral status with the methylome. And we build reference data set to profile the cell type compositions across these 156 samples. And we find out like CD8 T cells will be the main type of cells that getting increased in those people got latent CMV infection. Okay. Next questions is we were trying, since we find this, these ends like saying methylation and CMV infections is correlated, we're trying to know if we can predict the CMV seral status with the DNA methylation sequencing data we got. So to do this, we built a multivariate multiple linear regression models, which will be a systems of equations. So imagine here the Y matrix will be the methylation matrix. In rows, there will be every individuals. In column wise, there will be the 30K CpG size. We got a matrix shown here. And we have trait to measures. So for every individuals, we'll have different trait like age, sex, CMV seral status, cell type compositions, different ancestries, etc. in this trait matrix. And they will have a coefficient matrix, which will be response to per trait, how much they contribute to the methylation. Would be so this is the thing we could estimate, and then is the error vectors. We standardize the trap matrix from here, and then derive the coefficient matrix through the Penrose more inverse, or you can call pseudo inverse. So you can get from an idea from these matrix transpositions. So we have methylation, trait, and coefficient. So we can just move the pseudo-inverse trait matrix here, and we can derive the coefficient matrix. And then once we got the coefficient matrix, we can then predict the trait with the methylations and another round of pseudo-inverse for this coefficient matrix. Okay, so in this way, we can actually predict the trait with the methylation and the pseudo-inverse coefficient matrix. And you can see here is an example. We got coefficient scaled for every trait, for some of the trait we are interested in. And you can see some trait contribute really much correlated with the cell type, some really associated with age, and there belongs different CPG loci. So with these models, we can derive this predict an actual correlation matrix. So we put in age, sex, CMV cell status. We have lung and kidney cohort, although they are all the same biopsy, which is the blood cells. And we have principal components from cell types and also principal component for ancestry. And for each trace, we can predict them with the correlations above 0.4, which means these multivariate models, we can actually predict CMV serostatus with the methylation data we got. So here you can see the estimated values from CMV serostatus is lower in the negative serostatus negative patients and higher in those CMV positive patients. And you receive operator curve showing the area under curve to be 0.78, which means that this model, with the CMV epi scores, they do have the prediction rate for their true CMV serostatus. Okay, so now it's like we derive a model which we consider several traits inside this two cohort, and then to model them with DNA methylation, and we can actually predict the CMV serostatus with the methylation info we sequenced. Next, we are going to go deep into the locus-wise, like across of these sequenced regions, which CPG loci or what regions are actually more impacted by the CMV latent infection through methylation or not. Okay, so we use the multiple linear regression models to test for the associations, and we isolate several hundreds, we got hundreds of CPG sites and group them into whether they are hypermethylated in CMV positive patients or whether they are hypomethylated in CMV positive patients. So the sign is really important because usually hypermethylation is related to downregulation for that gene expression, and hypomethylation will be upregulation for that specific group of genes. So we do gene ontologies for this CPG loci. In the hypermethylated regions, you can see the terms popped out are more related to the neural systems. For example, it's neuroactive ligand, dendrite, or the assembly for synapse. Synapse is for neurotransmissions. So, and even the pattern specification. So the so here means for those hypermethylated regions related to downregulation of that group of genes, they are more related to neural systems, which means the neural systems get attenuated after CMV infections. Remember, we in the first slide, we know one of the features for congenital CMV infections that will include seizure, vision loss, and also hearing loss. They are all neural related, and we might find the mechanism through epigenetics, which means these group of genes, they are methylated after CMV infection. And for those hypomethylated regions, like related to upregulation of that group of genes, they are more related to the immune systems. For example, hematopoietic cell lineages, T cell receptors, MHCs, and also MHC complex. So, which is make sense because once the human get infected with the virus, their immune systems might goes up, which also showing up in those hypomethylated group of CpG loci. Okay. So here is an examples we found might be interested. So which is the locus in the second exon of a gene called ACE2, which is famous because it is the receptor for SARS-CoV-2, which which is catastrophic over the past few years, right? So we found there's 4 locus in these regions, in the ACE2 exon regions. These regions are hypomethylated. Color in orange would be the CMV positive patients, but more methylated in those CMV negative patients. So this is just an example showing like we are finding the locus regions that actually impact and showing the methylation distributions into different groups. So for these ACE2 regions, something is interesting is like several research have published like there's a feature called CMV and SARS-CoV-2 superinfection in vitro. This means like when a cell line they infect with the cytomegalovirus first, like this in the CMV will be colored in green. So for these, once the cell is primed with CMV infections, they are more easily to get infected by SARS-CoV-2. So it's like additive effect for these 2 viruses. So in vitro, they call these superinfections. You can see CMV negative and CMV positive group, they will have different rate to get the SARS-CoV-2 infected. Okay, and they even are trying to get the mechanisms to look into the gene, the ACE2 expressions. So you can see like the cells when they are primed with HCMV infections, the ACE2 expression is also going up in these immunofluorescence and also Western blot. They do quantitative PCR as well and showing once the cells get infected with CMV first, they are more easily to upregulated for ACE2. That's why they are more easily to get infected by SARS-CoV-2. So this is the bench work they do. So what I try to say is our finding in the ACE2 demethylation might be another mechanisms behind the SARS-CoV-2 and CMV superinfection. So quick summaries for now. We're trying to do locus-specific analysis, like to see across the human genomes which part are impacted by CMV the most. So we group these regions to be hypermethylated and find out from the gene ontology analysis, this group of loci are more neurosystem related. Since they are hypermethylated means they are more attenuated in CMV positive patients. And they might be related to the defect we see in congenital infections from the seizure side and hearing loss. And for those hypomethylated regions, they are more related to the immune systems like T cell activation, etc. And one of them is the ACE2 locus. So we found this locus to be hypomethylated, which might be a epigenetic mechanism behind the SARS-CoV-2 and CMV superinfection. Okay. So last, we're trying to verify the methylation data with transcriptomes. So we also got some of the transcriptomic data from the kidney cohort. So the numbers are listed here. So we're trying to find the differentially expressed genes between these 2 populations. We find out those CMV positive patients, those upregulated genes are more, you can see here, are more related to the T cell immune response, which is pretty inconsistent with what we found in the hypomethylated regions. For the downregulation genes, they are more related to oxidative stress and autophagy stuff. So we are able to derive some genes whose differentially expression between CMV positive and CMV negative patients might be through the differentially methylation. Okay, so it's integrated from those differentially methylated loci and differentially expressed genes. So we first do principal component analysis for these transcriptomes. We found for transcriptomes, the data is actually segregated or clustered very well based on their CMV serostatus. You can see those latent infected patients, they are more on the left side for the PC1 axis. For those not infected, they are going to the right of the PC1 axis. So we found the 3 genes really interested, and they are differentially expressed and also differentially methylated. So one is called BRD4, in short for bromodomain 4. So you can see they have bromodomains to bind DNAs, right? So bromodomain 4 genes, they are hypermethylated in the UTR of the gene locus, and they are downregulation in those CMV positive patients, which means these genes are silenced after the CMV infection. And other 2 genes called CD8A and CD8B. So from the names, you know, they might be they are part of the surface proteins for the CD8 T cells. And they are hypomethylated, which means they are upregulated in those CMV positive patients. Okay, so for BRD4, it's really interesting because a few years ago, there's a group of researchers, they found this group of genes might have epigenetic therapeutic intervention. Here they say bromodomain proteins, the main one would be BRD4 we just saw. So when the experiment they do is they treat latent CMV infected cells with the bromodomain. That inhibitors will attenuate the bromodomain proteins. So once the cells treated with this one, so the latent or inactive cells, they're starting to produce lytic immunogenes, which means they show to the host immune systems, they seems to become lytic, seems to become reactivated. However, they won't be able to reproduce their DNAs, and their viral immunomodulating proteins like the capsid to form virus particles is restricted, which means they show some signals to the host system saying, oh, this cells is infected by CMV, but actually they are not causing any damage. So people here saying they might be a good interventions like to evaluate for the host immune system to purge those latent viral reservoirs before doing any surgeries, for example, the transplant, to avoid those downstream immunosuppression effect like CMV viremia. So we find these genes also to be hypermethylated and downregulated in CMV positive patients, which says like DNA methylation might be an intrinsic surveillance that will tell the host immune systems like this cells is latently infected, and you can target these regions to remove those potential risk. Okay, so quick summaries for now is we found DNA methylation mediated genes, including BRD4 and CD8A and CD8B, these 2 CD8 surface protein components. Last is we're trying to make the DNA methylation data more more applications in clinical side. Is we're trying to do CMV viremia risk assessment with the DNA methylation profiles we sequenced. So we do this with the Cox proportional hazard regression. for the CMV viremia. So briefly saying the several risk for viremia is a really important risk assessment for patients about to going through an organ transplant surgeries. So you can see here D for donor, R for recipient. So the baseline would be those double negative for CMV serostatus. So which means they will be very unlikely to have viremia or infections after the surgeries. But for those patients, they were recipient negative but donor positive. You can see the odds ratio is really high. They might very high chance they will get CMV viremia in this group of patients. For those recipient already positive, there's way less chance to getting viremia than the high risk group. So we are trying to tackle this question to see if we can add one more risk assessment with the methylation in addition to the traditional several risk evaluation. So here we also group the patients into low serostatus, several risk, and with the recipient and donor both negative. The high risk group will be recipient negative and donor positive, right? So most of the patients, they will be intermediate. Remember, like we got almost like 60 to 80% of human populations are actually recipient positive, actually positive for CMV serostatus. So here is the time to viremia event occurring. So you can see the those low risk group, they remain no event happening to the baseline. For those high risk ones showing up here, they're really to 1 year they might got CMV viremia occurs. And we find out like those recipient intermediate group of serostatus, we can even stratify this with based on their CMV epi scores, which is the predicted CMV status with the multivariate models. With this record, we can see like the low epi score one, they go really easy to develop into CMV viremia. And those higher CMV epi score one will be in between of the two group of serostatus. Okay, so for kidney cohort, the samples is a little bit different, but you can still see the trend like epi score, the CMV epi scores can stratify these two groups inside those CMV positive recipient. So these tell us like CMV epigenetic scores we got from the multivariate models, they could serve as a covariate for clinicians or for us to assess those patients, CMV positive patients, whether they might get CMV viremia after they get the surgery. Okay. Finally, this is our models. So the color in the circles was based on the several risk, like a recipient, donor negative, recipient negative, donor positive into these two groups. We find, so these studies, we profile for the DNA methylations in the cohort, and we find the CMV positive patients, they might be hypermethylated in neurosystems and hypomethylated in immune systems. And all over this methylation profile, so we derive an CMV epi score per individual, per patient. So these scores can help us to do risk assessment for those CMV positive transplant recipient. So you can see the epi score serve like a balance over here for those inside this intermediate group, those subjects with less with lower CMV epi score, they got more high risk for getting CMV viremia. For those with higher CMV score, the risk will be lower in this group of patients. Okay. So this will be our final models to study methylation and CMV pathogenesis. finally, I would like to thank my mentor, Mathilde, and our lab members, Mila and Michael, and our collaborators in Reed Labs, Elaine, Joanna, and Harry, and also the Greenland Labs in UCSF, John and Rashmi, for their help for those clinical samples and also cohort recruitment sampling. And thank you for joining the talk today, and happy to take any questions.
Thank you, Fei-Man, for a wonderful talk. Any questions?
Yeah, I have a question, just quick. So was there a reason why you didn't directly model the traits rather than you do it the other way and then you invert the matrix?
You say why we have to invert?
The multivariate model?
Yeah.
You wanted to get the prediction of the trait?
Yeah, so I guess this model is like we do several ways. We present this one is because we wanna, since it's the clinical studies, so we have to model every trait at the same time to look into their contributions per CpG loci. So that's why we trying to model with multivariate model.
Well, what I'm trying to say is trait matrix could be on the left-hand side and matrix could be on right-hand side, and just directly modeled that way. Why was it the other way?
You mean like we put trait?
Yeah, usually traits or phenotype matrix, a methylation matrix could be, in genetic setting or a lot of regression setting. You could do it.
We actually do that per trait with the methylation. Yeah, but I think is the traits, they might be also have correlations among of them. So it's better we model them at the same time to have the final predictions.
I guess trait is outcome and methylation is on the right-hand side. So you just interchange M and T matrix. That is so multivariate model, adjusting for the correlation between the traits. I guess in this field, this is the convention. This is how you model.
Okay. Yeah, I understand your question. Is overall I think is like for the pseudo inverse method we're trying to propose is because it's really easy for computation, and we can get this coefficient matrix one time instead of doing the per site and every trait together one by one. That's the thing.
And follow up on that, how large are these errors and how do they affect this one?
I don't quite remember the error, but I would say the prediction from the trait to the scaled values is pretty close in this multivariate model.
Is there any bound when you model this?
Bound?
Yeah, like the upper limit on the errors that is acceptable kind of thing.
No, I didn't do that, but I'll consider that. Yeah.
Okay. Any further question? If not, let's thank Dr. Fei-Man Shu for the wonderful talk, and hopefully I'll see you in our next presentation next month, and wish you a good rest of the day, and thank you for joining.
Thank you.